Tammy, I'll have to mute you for now. Okay, fantastic. I think we are good to go. Hi, thanks everybody for joining us today. As you know, we all love compliments, but founders are built very different. They are not big fan of sugar-coated stuff like, hey, interesting idea, or let's catch up soon. They actually crave honest feedback, tough questions, and maybe a purchase order. That's where EthAum comes in. Our roast sessions are designed to turn up the heat live and unfiltered. Founders face tough questions, real feedback, and zero fluff from seasoned experts. Think of it like a Shark Tank, but less drama and more insights. Today we have Marco from Peptitech stepping into the spotlight. Marco is going to present Peptitech's solution, answer some difficult questions, and get real feedback from our diverse and really awesome panel. Let's start with the introduction of our esteemed panelists. I would first request Vasileios to go and introduce himself. Vasileios, please.
Hello, nice to meet you all. I am currently working as a Chief Operating Officer at a Metropi Eye Clinic in Crete, along with Professor Aslanides. And my background is mostly on computer science and AI and medtech. And I am chief of research also in our department. I have several big experience also with some European grants also and things like that.
Fantastic. Thanks a lot, Vasileios. Really appreciate your time today. And I'm sure that we will have a lot of really interesting questions and insights from your side. So thanks a lot for joining us today. Dr. Saad, could you please introduce yourself? Please unmute yourself.
Okay, I'm Dr. Ahmed Saad. I'm an ophthalmology consultant. I'm MD, PhD, and I work on the academic clinical ophthalmology work. I have multiple publications on ophthalmology researches regarding the new devices evaluating the corneal biomechanics, which I performed in World's Eye Hospital, where I stayed for my cornea fellowship at World's Eye Hospital, Thomas Jefferson University, Philadelphia. And I'm where I have the good pleasure to have a fellowship in such this prestigious hospital, which is ranked among the top 5 hospitals in the world or in the United States in ophthalmology care. I'm interested in researches regarding new devices, new innovations, or new medications for the retinal problems or eye problems, where we consider it as a challenging cases or challenging diseases, and we try to give a hope for the patients with these conditions.
Thank you. Thanks a lot, Dr. Saad. Appreciate your time today. Tammy, could you please introduce yourself? Yeah.
Hi, I'm Tammy Movsas. I'm by training, I'm a pediatric neuro-ophthalmologist. I have interest in brain and eye neuroprotective mechanisms. And I'm also a epidemiologist, and I also have a startup company. So I'm coming to you with multiple integrative expertise.
Fantastic. Thanks a lot, Tammy. Really appreciate your time today. And now we have Marco, founder and CEO of Peptitech, joining the session today. And obviously, Marco, I would request you to introduce yourself and then get into the Peptitech presentation so that we can get a lot of interesting questions and insights from the panelists. Marco, please go ahead.
Okay, thank you very much. Thank you so much to have this very various panelists, and I think it's going to be very interesting talking to you about what we are doing. I'm a professor, associate professor in pharmacology, and I'm working in neuroscience field from many years. And I'm a preclinical pharmacologist researcher working in the field of neurodegeneration And recently, thanks to Peptitech, basically, I started to work also in the retinal area because, as we know, retina is part of the brain. And the neuroprotection is actually what I'm trying to achieve with this project, and among many other projects that I'm working on in this moment. As Pankaj was telling us, so I would like to present to you what we are doing as a Peptitech. So I founded Peptitech a few years ago because Peptitech is working on the development of a biotech platform targeting neurodegeneration. And as a first indication, we want to address retinal neuroprotection in glaucoma. You all know that neurodegeneration is basically still an unmet medical need because we don't have a real disease-modifying drugs. So as we all know, glutamate is a neurotransmitter very important for the brain, is the excitatory neurotransmitter mainly represented in the brain that is important for the physiology of the brain, but also when it's overflowed, when it's released in excess, is causing neurotoxicity, leading to several pathologies spanning from the neurodegeneration to retinal diseases as well. And we don't have any approved disease-modifying therapies targeting this mechanism. And basically, the current drugs only manage symptoms, but not are able to change the pathology. We have, therefore, a massive therapeutic gap for selective neuroprotective agents for addressing the CNS diseases. Actually, the current approach is to inhibit NMDA receptors. And this, of course, is working for preclinical models, but blocking these channels means blocking all the neurotransmission. And therefore, this is compromising the physiological activity, giving a lot of side effects. And we know, for example, memantine is used in Alzheimer's disease, but gives a lot of side effects and very, very difficult to be used, actually. And memantine is basically one of the only neurodegenerative or NMDA inhibitors that has been approved by FDA. What we discovered is a new molecular mechanism of action, which is actually underlying the NMDA receptors. So basically, when we have a toxic insult from the presynaptic NMDA receptors, we have an activation. And what we discover is that the release that is activated by the toxic insult of the NMDA is driven by the interaction between these two proteins at the presynaptic side. And this is regulating this high level of glutamate released. We basically designed a cell-permeable peptide of 26 amino acids, which is able to reestablish the physiological level of glutamate released without affecting the pre-synaptic NMDA receptor, and absolutely doesn't affect the postsynaptic NMDA receptor itself. And we patented this, and now what we are working on is retinal diseases, because we know that retina is considered a window on the brain, a window on the CNS, basically, and the retinal ganglion cells are part of the CNS because they are CNS neurons, and they degenerate also due to high level of glutamate release. And so far, what we know is that hypotonizers are the drugs used for glaucoma, but still the 40% of the people that are treated get blind from one eye, or 16% of them both eyes. So we have a gap because we don't have neuroprotective agents for this market of retinal degeneration. We published several papers, and many other groups has already reproduced our mechanism of action. And lately, this year, we published, actually a few months ago, 1 month ago, we published a new paper in which we were validating the use of our peptide as an eye drop for protecting the retina in few mouse models of retinal degeneration. We basically, why we think our peptide is the first-in-class solution, because it's effective as an NMDA, let's say, and affecting reducing the NMDA evoked release on the retina, is able to block the JNK2 syntaxin 1A interaction, is able to do this as a topical administration. So we just need eye drops. And is able to reduce the glutamate release overflow, and this is able to reduce the apoptosis. So therefore, we think this can be a suitable solution for several pathologies. In glaucoma, for example, we know that in these glaucoma mouse models that we have in our lab, we have an increase of glutamate, as you can see here, when we have inducing this mouse model. And we know our peptide is able to reduce this. I cannot show you this data right now because they are not published. But what we think is that our peptide can become an add-on therapy on the current therapy as a hypotonizer eye drop. So can be given as an add-on therapy to contrast blindness. As we know, blindness is a very social, a large social problem. Several billion of people have this vision impairment due to retinal degeneration. We have several worldwide people, as you can see here, for different pathologies that are facing blindness. The expenses is really high, but also the market is very high. So we will have this projection, for example, that show to us that the market is going to increase a lot in the next years. In 2030, for example, we have this increase. So basically, Peptitech wants to build a platform because we are working on both eye pathologies and CNS pathology, because these pathologies are basically sharing the same mechanism of action that we are addressing with our peptide. So far, we completed the discovery, and we are in preclinical phase with this peptide for glaucoma. Thanks also to some fundings that we got, business angels investments, and so we have efficacy in vitro models, in 3 different mouse models, also in glaucoma. And we are also we are applying also to European grants. And this is another part of our platform, because I applied recently to with this large consortium to an AIC Pathfinder in order to validate a novel biomarker for glaucoma as a glaucoma intraocular sensor, in order to check whether the glaucoma, so whether the glutamate is basically released in excess in the eye. We also have de-risked a portion of our project thanks to a partnership with Evotech and this consortium Extend, in which we did several experiments, and we basically validated our peptide for epileptic pathologies. But of course, the epilepsies cannot be addressed with a peptide like ours. So what we think that our platform will be composed by this peptide that we are working on right now for retinopathies. a novel small molecule based on the same mechanism of action for the CNS epilepsy, and we are already starting to think and working on AI-assisted drug development for that. And also this biomarker as a glutamate intraocular sensor. So these three are becoming our pillars for the next future, starting, as we said, on glaucoma as a first indication. So to work in glaucoma, we are fundraising because we have a drug development pathway that we have already designed, and we are looking for 800K as a seed phase activity. EthAum, that is hosting us with this nice event, is in soft commitment for this. And we think in 1 year and a half, we will be able to come to close to the IND enabling to close the IND enabling studies to get to the clinical phase 1 with approximately 3 million of investment. We have a large scientific team, myself as a founder, we have a preclinical R&D, head of a preclinical R&D, ophthalmology senior scientist, and also we have people from the business portion of the business partners that can help us with the Peptitech. And yeah, I'm happy to talk about our project with all of you. And these are my contacts eventually if you want to contact me.
Thank you. Thanks a lot, Marco. That's a very quick presentation from Marco. And now, obviously, he's ready to answer any difficult questions. Right, Marco?
Yeah.
Okay, fantastic. So who wants to go first? Yeah, Tammy, please go ahead.
Okay, I'll have a whole bunch of questions. I hope that's okay.
Please.
Very good.
Oh, okay.
Very good.
One is, okay, well, to me, one of the key questions, I had sort of reviewed this ahead of time, so I wrote things, I took some notes ahead of time, is that well, first of all, the science is intellectually elegant. So I'm very interested in it from that point of view. I have concerns about it being an add-on drug to glaucoma. What part of the reason is that there's the expense of that, because eye drops, you're saying it's not instead, it's in addition. And a lot of people do well with eye drops. So what would be the actual treatable population, and how would you identify who needs this particular drug as an add-on, as opposed to saying it's for all of glaucoma?
Okay, let's say that, of course, in my presentation, I was citing that we are also working on a biomarker, potential biomarker, which is telling us basically when it's actually becoming important and when the glutamate is really released in high level and toxic level, and that is inducing apoptosis and cell death. And of course, this is an embryonic project. So of course, it will not be ready when we are selling our product to the people. But already on the market, there are some very interesting biomarkers. One of them is called DARK, which is actually in clinical phase 2 and is actually used already from some other companies. And actually what they do is an injection of annexin 5, and annexin 5 can go into the retina and tell us when the cells are starting to go to apoptosis. And this, I think, is the right time to give our peptide.
I see.
To start the neuroprotection, you know.
Okay, so in other words, it would be add on to people who have been identified as high risk.
May I add a notice here, please?
Sorry?
May I add a notice here, please?
Yeah, please.
Yeah,
Sure.
yes, of course.
From my point of view as an ophthalmologist, we can learn or we can know how if the ganglion cell layer of the patient of glaucoma is markedly affected, or how far is it affected? It's affected by doing several investigations for the patient, like perimetry or visual field examination, which examines the function or the functionality of the ganglion cell layer, or how far they are working. Moreover, we can detect the anatomical or the loss of the thickness of the ganglion cell layer by doing spectral domain spectral domain OCT, or optical coherence tomography, to measure the ganglion cell layer thickness. So we can learn here, or we can know here, how far his ganglion cell layer is affected. So he can be like a candidate for this medication or add-on medication, or is not suitable for that. Thank you.
I see. Okay. Thank you. If
And then I have to say, we have another feature of the peptide which we cannot say right now, because it's it's not disclosed right now. So it could be eventually used as a alone.
Prophylactic treatment. Yes.
Yeah, prophylactic.
Okay. I'll ask one more that other people ask. I actually have like 10 questions here, but I'll ask you one more for now. So I'm actually less excited that it's an eye drop, because I've actually myself have tried to figure out ways to get the eye drops in. The patients have so much trouble with the eye drops as it is. And one big reason for glaucoma failure is actually just getting the drops into the eye. And there's over 100,000 patents right now of devices to try to get eye drops into the eye. And people are working on trying to mist eye drops into the eye. And there's some trials going on with that. So one is, first of all, a lot of people are on multiple eye drops. So now we're talking about maybe adding, I guess there's so many issues with eye drops about whether or not it really goes in the eye, if they're really waiting the 5 minutes, if they're occluding their thing, if they're able to get it into the eye, if they're willing to use more than one drop. So I don't know if there's if you've thought about a different way, if it could be misted into the eye, if it could be maybe put as a pellet, and sort of slow release.
Yeah.
But I would be more excited. Adding on another eye drop to me is like already a problem, because part of the reason the eye drops don't work is they can't get them in the eye for whatever reason. So.
Oh, this is a very good point, actually. So I have to say that still we are not working on the formulation because of this reason. So we would like to be sure to understand very well from ophthalmologists what can be a substitution of eye drops. For example, lenses with slow release. For example, there are some injection that you can just leave it in the eye forever, something like that. But so I mean, to answer your question, yes, we are also trying to understand, for the compliance of the use of this potential drug. So what is going to be the best administration route, or what is going to be the best, let's say, formulation for that. But I have to say, so if you were working in preclinical studies, imagine how difficult it is to tell a mouse to keep the eyes open with an eye drop. So it's very difficult. So what we did...
Yeah, I've done that. I mean, the thing is, though, you could get it into the mouse's eye. It goes in. I mean, with people, it's...
No. Okay, so what we did with the mouse is just to keep the mouse 90 seconds with a drop of our peptide on top of the cornea, and keep it the cornea. Right. I mean the eye open, and keep it open for 90 seconds, no more than that. And the peptide is so cell permeable that can get inside, and 1 drop per day is enough to reach the concentration that leads let's say, leads the effect. So we have the effect after 1 week. We basically can see already the effect. So I mean, to answer you are right. We can work on the formulation.
I would like to invite Vasileios. Yeah, please go ahead.
Another issues continue from where Tammy stopped is not only that you're not very sure 100% that people with glaucoma are really putting correctly their eye drops in their eyes, is even themselves after some years they get too tired of it. And there are in the literature there are several studies that show that about 30 to 50% of people are not compliant with their medication. So adding perhaps another drop on their everyday regime will be an issue. Perhaps a solution will be either a kind of different kind of formulation, as you said, like a slow release drug, a lens or an implant, something like that, or a combination with another medication like prostaglandin or something like that.
Exactly.
I don't know if you have already studied how your product, this peptide, is working combined with prostaglandin. If there is
no,
any
we didn't do these experiments already, but of course, as you said, potentially a pharmaceutical company can put together these two drugs in one drop. So
there will be some studies will be needed to see if this
of
kind of combination is working, if how effective it is.
Of course.
And I think, or for example,
additionally, we can use this drug as an intraocular implant, like we have in the market as ophthalmologists, anti-glaucomatous medication, which is introduced as intraocular implant or intravitreal implant. We can
like a Nosurdex, I think, or something like that.
No, it's not Dexa. It's like a prostaglandin analog.
I mean the same way of delivering the drug.
Yeah. It's like
putting in the eye and then slow releasing it.
Slow release implant of the drug inside the eye. We can use it even as a subconjunctival injection or subtenon injection or preocular besides the glow injection. But to be as an eye drops, I believe this is a great achievement because some people are not reachable for glaucoma surgical management or intervention, like patients in territories or at different countries, which are or they found like difficulty in moving to the hospital. So to get the intervention for their glaucoma medication or glaucoma management. But with hello.
Yeah, we can hear you.
But with the eye drops, I believe it will be reachable for all the patients, and it can be like tolerable for the most of them. I know that some patients may like complain from tolerability with the medication in the form of eye drops because they are not like well trained how to use it. They may miss doses of the medication or miss some drops outside their eye, but it's much better than a loss of the whole medication because it should be introduced as an intravitreal injection since we have in the retinal diseases management like diabetic retinopathy, like diabetic macular edema, like vein occlusion, we should introduce the drug inside the vitreous body or intravitreal injection, not to be an eye drops, which is anti-vascular endothelial growth factor medication. But to be as an eye drops here, it's a great achievement.
For sure, if it can be as an eye drop, it will be readily available and easily accessible from the majority of people. The thing is to make sure that they are going to be compliant with their medication.
Yeah,
exactly.
That's why I think if it can be combined with an already existing anti-glaucoma drug, and instead of having another drop, just keep the same number of drops you are using, it will be much better.
We have many like medications for glaucoma, which is combined, or which are combined medications like beta blocker beside another one like Cdamax or like drugs working on different pathways, not on the same pathway.
Perhaps
I think this can be a very nice solution. The combination, and also maybe to have multiple potential, multiple combination or also formulation. Maybe it could be even we could all even think that in the future, of course, this drug could be also injected in the eye for some patients, or other can have eye drops.
Different versions of it.
You know, different versions. Yes, yes.
Thank you.
Okay.
And of course, different concentration as well. Yes.
Exactly. Yeah.
We can take more questions, obviously. Yeah, Teresa, please go ahead.
For your non-glaucoma indications, are you planning to continue with the eye drop, or would it be a completely different formulation and presentation?
For non-glaucoma, you mean for the CNS?
Uh-huh.
Yeah. No, for sure, for the CNS. So we had already, as I said, the risking experiments done. So we basically know that the peptide is able to reduce seizures for epilepsy, for example. But of course, I think it would be better to have small molecules instead of peptides. So what we are doing is to try to get new small molecules able to interact with this molecular pathway. And therefore, it could be, you know.
I think such an approach would be more beneficial. Perhaps I'm not in your expertise, in your as an expert as you in this kind of area, but as far as I know, peptides are also very unstable, and it's not very easy to keep them in the temperature. They're very sensitive, and how they are going to stay active and as they should, being on a shelf waiting for someone to buy the product.
Actually, our peptide we tested already and is very stable at different temperature. So can go from -20 up to 90 degrees and is very stable. But of course, is less stable in enzymatic environment that could be a systemic injection. No, so in the body, for example, for the eye, there is not such a big problem because the eye is a very
closed,
let's say closed and system, and basically also the drugs that you are insert in the eye is not withdraw in a very is not deleted very easily. But of course, a systemic injection of a peptide is not the same efficient as a potential a small molecule. That's why we are thinking a small molecule for a systemic use.
So would that small molecule be effectively, we'll call it a synthetic peptide with non-natural amino acids, or is it going to be completely different?
Yeah, this is another option that we are also trying to address. This is actually this is the basically the idea to produce other peptides with as you said, so amino acid that cannot be addressed by enzymes. But also small molecules, and I'm thinking of molecules like synthetic molecules, you know. So starting thanks to AI assisted software, we can test different molecules like libraries of molecules and select one of them in a funnel way. And find out if we can, find the right molecules able to disrupt this interaction between JNK2 and syntaxin-1A.
Sir, Dr. Marco, concerning the concept of the way it acts or it works on the ganglion cell layer survival or protection, I believe we can, or you can try it on different pathologies in the eyes or different conditions that may affect the ganglion cell layer, not only glaucoma, but also you can use it in like renal ischemia, like optic nerve ischemia, like intraocular inflammation, like which is inflammations that damage the inner retinal layers, like endophthalmitis, like panuveitis. I believe it will work on the same way by protection or by protecting the ganglion cell layer from death due to the condition that the patient has.
Yeah, this is a good idea. And actually, I mean, in our development plan, we are not thinking only to try it for glaucoma, but also for other pathologies, and for sure retinal ischemia, or even diabetic retinopathy. It could be one of the indication. Yeah.
Yes, exactly. Yeah, yeah. Like because we face, or we have a lot of conditions like ischemic diabetic retinopathy, like ischemic optic nerve lesion neuropathy, and patients with central retinal artery occlusion where their central vision are lost permanently, and it is not like managed or treated after loss of the ganglion cell layer by any way.
Yeah, yeah.
Okay.
I think Tammy had quite a few questions. Tammy, could you please go ahead?
Yeah, so I guess sort of related to what you were just saying, why did you sort of pick glaucoma as its first sort of indication to try? Because the glaucoma trials are very long, vision losses, slow, already receiving, we have all these other potential indications, which there is nothing else, and that the vision loss might be faster in general. So I guess, wouldn't it make more sense to pick one of those other indications as opposed to glaucoma as the first indication for this drug?
Oh, I mean, we started from glaucoma because it's one of these pathologies in which the glutamate is absolutely pervading all the retina, So and it's actually very intuitive, the fact that while the IOP is increasing, the mechanism to which the glutamate is released is because of also the IOP increase. So actually talking to ophthalmologists, the clinical trials shouldn't be very long. So I think in 1 year you can see whether the pathology is worsening or is not. So I think it's, and also because the market is very large, so many people has this pathology, especially in Asia. There would be 50% of the patients worldwide. So it's a huge market as well.
Yeah, well, the market's not quite as used because you'd have to have the addressable market. We sort of talked about that. Like when you take some of these other indications with there's nothing else, the addressable market might, be a little more desperate. Okay, let me just. Okay, I think we talked about some of what I was going to ask you about degradation. You talked about that, about it's a large peptide. So just sort of getting down to your patent, the umbrella patent was filed like in 2014 or 2015. How much left do you think? You sort of said this is still in its sort of embryonic phase. How much longer by the time you get this approved for use, what do you expect the patent?
No, actually, the patent will be renewed in probably in few months, because we have new characteristic of the effect of the peptide that will allow us to make a new patent. And also, as we were talking about before with Teresa as well, so we basically want to maybe to have another patent by changing, the amino acid with non-natural amino acids. So maybe to increase the efficacy, maybe to increase the let's say the stability of the peptide for CNS uses. So it can be, renew the patent.
Okay, so you'll have more IP. I guess you'll have additional IP as the years go on.
Exactly.
The other thing is, you sort of said that this molecule sort of gets activated under stress.
Yeah.
How are you going to show sort of in vivo that it's not being activated just under physiologic conditions? And if so, would that have any effects?
Oh, because we under physiological condition, of course, we have an activation of this pathway, but is a feedback is a back feeding activated mechanism. And if you see the image that I was showing to you before, I will show it again. And basically, let's see if I can. Yeah, can you see it?
Yeah, we can see your screen.
Can you?
Yeah, we can see your screen.
Yeah.
You can't?
We see your screen, yeah.
Ah, you see it. Okay. So basically here, yeah. So as you can see here, so the glutamate is increased in the retinal tissue, in all the retinal tissue in these glaucoma mouse models. And if we are looking at the interaction between JNK2 and syntaxin 1A, and we did this with a PLA assay, so a proximity ligation assay, we see here a huge number of interacting proteins together instead of in the physiological level. And also we can show, and actually we published this already, that if you don't activate the NMDA receptors, you don't have an inhibition of this pathway if you use the peptide. And also what we have shown is the peptide itself is not able to interact with the channel, NMDA channel, so it doesn't affect the opening of the channel, and calcium can still flow into the channel.
So you would show that sort of clinically by giving it to people without glaucoma, the fact that, like, how would you sort of show it has no...
You mean in clinical trials?
Yeah, like in a clinical trial, how would you show that it doesn't have sort of side effects if someone doesn't have a lot of glutamate?
Yeah, of course. We're going to have an arm of these people without, with no pathologies, eye pathologies. And
so you'll have to
show, and we're gonna show that there is no effect, no toxic effect on the retinal ganglion cells.
And,
I see. Okay.
Sorry for stepping in. Also, because you have to define also somehow. I don't know if you have already done it. The dosage finding strategy and the safety margins that there should be there.
Yeah.
As far as I saw in your recent publication, because I did my sort of homework before that. There was like a dosage of 500 microns, I think, that lowered the glutamate below the baseline levels.
Yeah, which is a bit toxic in this case. So basically, what we are working now is with 25 micromolar instead of 500. Because 25 micromolar, and we are working between 10 and 25 micromolar.
Okay. This has to be very, especially if you go for FDA trials and things like clinical trials. This has to be well defined, the dosage strategy and what are the safety margins.
Yeah.
So Marco, I have a question here. What type of glaucoma you have tried the medication or this innovation on? Like glaucoma has different types, like different like forms, even acute or chronic, and each one has different subtypes. So I'd like to see, which type did you try this medication?
So now, for the moment, we tried, as we said, so we are in preclinical trials, so we tried on mouse. And what we did is injections of NMDA for 1 month, which is a type of kind of chronic degenerative disease for the retina. We are also trying, and this is already published, but what is unpublished is what we are trying now. So the injection of Cephalose beads with hyaluronic acid on top. And this is increasing the IOP of these eyes up to 15, 14, 20 days.
You create a simulation of increased intraocular pressure by injecting fluids or liquids inside. And
injecting beads, which is sticking into basically are not allowing the anterior chamber and the posterior chamber to communicate. So basically the fluid exchange is blocked, and so the IOP is increased.
So to increase the intraocular pressure inside the eye. Because we have a subtype of glaucoma, we call it, or we term it as a normal tension glaucoma.
Exactly.
Where the patient's blood pressure is within normal range by all textbooks, but he already lost progressively fibers from retinal fiber layer, which is not surviving with this normal pressure, which is nothing to do for him to lower his pressure to manage him. So the best management here in these conditions are the neuroprotection. We used before beta agonist eye drops, but it's working on increasing the blood flow. Not or its main effect is increasing the microcirculation blood flow for the nerve fibers, not to protect the ganglion cell layer, or its sub or sideway effect was a neuroprotection, not mainly the effect is a neuroprotection. So I believe that this drug will be the best medication for these conditions. The patients with normal tension glaucoma.
And the NMDA injection is mimicking such type of glaucoma because it's inducing the degradation, let's say, neurodegeneration of the retina without increasing the IOP.
Exactly. So here, these conditions, even with a normal blood pressure, the nerve fibers can survive. So so the prescribing medication or prescription of anti-glaucomatous medication or lowering the intraocular pressure is not the solution.
Although is used in clinical.
Sure.
Practice.
In the market, or in our clinical work. But it is not that, or not such a potent effect or potent medication for protecting the patient from progressive loss of his nerve fiber layer.
In your animal studies, have you done anything in the systemic injection arena?
So we did publish already a paper in which we injected the peptide in animal with in normal animals, wild type animal, and we found that the injection leads to leads the peptide to reach the brain within 2 hours, and it stays up to 24 hours. So we still see the effect in 24 hours. In that case, we were taking the brain and doing the immunoprecipitation of the two proteins, JNK2 and syntaxin 1A, and we saw that they were losing the contact among them. So this is something that we have done. We did then some experiments in epileptic seizure mouse, and we saw some very preliminary data in which the peptide was highly, highly capable of showing the reduction of cell death in these animals. So we have some data on that. We did around 20 animals per treatment. So, but I mean, in that case, we were injecting the peptide directly into the brain. these animals. But the effect was really striking. That meaning that the molecular target is really important in this pathology. And actually what we think is for the epileptic seizures pathology, so we think it would be important to use this in the secondary epilepsy. So meaning, so it's very difficult to predict when a patient, when a subject will go to have epilepsy. So, and also the cell death from epileptic seizures is supported by the increase of the concentration of glutamate after the reduction of GABAergic system. So the glutamate system is let's say increased, and therefore the cell death is coming from this kind of glutamate increase. So we think the peptide could be, or let's say a drug, a small molecule would be absolutely useful for the secondary forms of epilepsy. So when we know that this patient has epilepsy, so to neuroprotect this patient, we can use it as a prevention.
But at least within the animals that you've worked with, you haven't had any toxic impacts of systemic.
Absolutely no.
Okay, good.
None of them. No.
No,
neither for the ophthalmic application. So we never actually. Sorry, Dr. Hamed. So we did once. We tried in rats with induced diabetic retinopathy, and what we saw is that the eye that was treated with our peptide didn't get this kind of cornea, modified cornea that normally these animals has. So the eye in which we were putting basically using our peptide was protected somehow from changes in this pathology. Yeah.
Mm-hmm. Yeah,
I think Dr. Saad,
yeah, we have some cases when treated from ophthalmological condition, the management line or the treatment line itself has a toxic side effect on the retina. Have you tried this like medication in combination with these other medications, like injecting antibiotic inside the eye for intraocular inflammation or intraocular infection? The antibiotic itself or the preservative exerts some toxicity on the retinal layers. And
no, we didn't try. No.
Yeah, and for patients treated with argon laser for retinal conditions.
Okay.
Applying the retinal coagulation with the laser beam, some ganglion cells are lost during the application of the argon laser. Didn't you even tried this medication besides this line of treatment, because it will be changing the prospect of the treatment of the patient?
No, actually, we didn't try this. So we didn't try the co-treatment with these drugs. But what I can say is that in our last publication, we were finding that the peptide is able to reduce potentially the inflammatory response in these animal models. So we saw that there was a reduction of inflammatory, like a caspase signal, and therefore we think that we can even see a reduction of inflammation. And this is due to the fact that junk proteins are activated also in during inflammation. So being our peptide a junk2 inhibitor, that might be the case.
Mm-hmm.
Thank you. We are top of the hour panelists, so really appreciate the conversation so far. I just have one request. So let's say if you are a guide or advisor or mentor of Marco, what is the one advice that you will give to Marco? Just one key takeaway, one guidance, one insight to Marco to run with it. Who wants to start first? Tammy,
I could start. Yeah, I would say I didn't ask, but I take it that so far everything's done in rodents like mice or rats. I would sort of say like we should at least validate this in more than one type, as opposed to like you said you did 3 different types of retinal degeneration in one animal type. I would like to sort of see that this is could be expanded to rabbits and higher mammals. Because as we know, a lot of things work in mice and they don't necessarily, even though I think it's very elegant and I'm very hopeful for it, but I think we need a little more data that it works in more than just one animal type.
Yeah, thank you very much.
I have to jump off now, but thank you for the opportunity.
Appreciate it.
Okay.
Yeah.
Bye-bye.
Okay, bye-bye.
Vasileios, any last comment from your side? Any last guidance, piece of advice?
Yeah, well, I would say, as we discussed before, to underline the opportunity and the potential ability of combining your peptides with existing antiglycomatic drugs, and make sure that you either already have or you are going to have a well-experienced team in regulatory affairs, because it's going to be a huge burden going from the clinic to the market. When you're going to start attracting some funds and asking for things during fundraising, they are going to really need to know that they know what you're doing in your team.
Thank you very much.
Thank you. Thanks a lot, Vasileios. Dr. Saad, please.
Yeah, my advice is to keep going for this great innovation or great medication, because it will give a hope for us and for the patients as well. For managing cases where we were considered, it's like it has no way to manage anymore, or no help we can give to the patient anymore. I believe it can be a potential, great source of help. Yeah.
Thank you so much.
Thank you. Thanks a lot, all the panelists. Again, really appreciate your time today. All the questions, all the insights are extremely helpful, and I'm sure that it will help Marco fine-tune the overall strategies, the product roadmap, the differentiation for the company in the near future. Marco, what you did today is extremely commendable. You invited criticism for yourself on a live stream, on a public forum, and responded to all the questions very positively with a smile on your face. So really appreciate that. Hats off to your guts and positivity. Not many people can invite a roast session for themselves in such a setting. So really appreciate that. It's truly inspiring. Hope this session brings a lot of new energy, ideas, and growth trajectory to Peptitech.
Thanks. Thanks a lot.
Thank you, everybody.
Thank you very much.
And we'll reconnect soon. Take care. Thank you.
Bye.